Good evening, everyone. Thank you for joining us today. My name is Amanda Brito del Pino. I'm a member of the Coalition of the Americas. I am working in our relations with the US market, with the US and Canadian markets. Today, Violeta and I were invited to lead this presentation in the framework of the World Hemophilia Day. There are a series of meetings in order to keep everybody up to date in terms of treatment trends. There are several industry leaders. There are companies such as BioMarine and Pfizer. We can choose from wherever we are to provide. I'm going to give the floor to Violeta, who is going to introduce Dr. Boadas. Thank you. I am Violeta Farah. I'm a coordinator in the Youth Committee. I am from Venezuela. I am a member of the Hemophilia Society in Venezuela. It is an honor for me to introduce Dr. Boada, who is a very good friend of ours here in Venezuela. She is a physician from Venezuela with a postgraduate in internal medicine and hematology. Until July 2021, she was a private consultant in the Capital District Blood Bank. And since August 2021, she has been the head of research and teaching hospital of the Municipal Blood Bank in Caracas. She is a postgraduate professor in the Central Venezuelan University, and she is a member of the CLAHT group, and ISTH and ISH. Dr. Boadas, you can take the floor. Thank you both. Thank you all for inviting me to participate in this pre-celebration of the World Hemophilia Day. We are going to talk about extended half-life concentrate factors. Let me introduce the factor 8 molecule. This is a protein which is quite complex and big. It has 2,332 amino acids, and it is a heterodimer with one light and one heavy chain. Its half-life is 8 to 12 hours. As to factor 9, it is a hepatic synthesis or liver synthesis serine protease. It is a glycoprotein of 415 amino acids, much simpler and smaller than the other molecule, and its plasma half-life is 18 to 24 hours. This is a timeline of hemophilia treatment. By the mid-1970s, treatment started to improve. This is the beginning of modern hemophilia treatment because in the 1970s, we started using plasma concentrates for hemophilia treatment, and we started using desmopressin as well. Subsequently, in the 1980s, these plasma concentrates were treated for viral inactivation. So these are safe concentrates. Also around the 1980s, and even earlier than that, since the mid-70s, multidisciplinary treatment was implemented. This is a blood condition that affects joints, muscles, the entire musculoskeletal system. So it was necessary to include physiotherapists and orthopedists, as well as pharmacy, psychology, and many other professionals in the multidisciplinary team in order to improve patients' treatment. By the mid-1990s, production of recombinant factors started, as well as bypassing agents began to be developed for persons with hemophilia who developed inhibitors. Afterwards, by the year 2000, we had the technology and bioengineering for extended half-life concentrates. So the goal of prophylaxis in hemophilia A is, you know, that prophylaxis is the regular infusion of missing concentrates, whether factor 8 or factor 9, whether it's hemophilia A or B. So in regular prophylaxis, the infusion of these concentrates is used in order to prevent spontaneous bleedings and, of course, avoid joint damage. It was said that a 1% factor 8 level was considered the goal for prophylaxis, but after 2020, with the release of the World Federation of Hemophilia guidelines, the WFH realized that 1% was not enough. There was still joint damage when using 1%, and that is not what was desired. So they started recommending 3 to 5%. This has grown, and what they try to achieve now is to have 0 joint bleeds per year. And this would be with 10 to 15% of factor 8 in the case of hemophilia A. We see that pharmacology research has 2 main lines of research. One is the development of new molecules to improve the characteristics of drugs already available. And the other is to research changes in the mode of administration or modifications of some original molecules in order to change and improve the efficacy and tolerance of these drugs. The pharmaceutical, the prodrugs, the sustained release dosage forms, and association with lipid forms are some examples of how you may improve these molecules. The half-life of a drug is the time it takes to eliminate 50% of a drug or medicine. So half-life in the case of coagulation factors determines how frequently these factors should be administered in order to protect the patient against bleeding. So hemophilia A treatment requires factor 8 concentrates, and each factor 8 unit infused intravenously is going to increase the plasma level of factor 8 by 2%. Standard half-life concentrates last 8 to 12 hours, and prophylaxis needs to be performed 3 to 4 times a week. With extended half-life concentrates, half-life is approximately 19 hours, and prophylaxis is required twice a week. So prophylaxis is used for patients with severe hemophilia, with a severe phenotype. Usually those who have under 3% factor. It is a continuous weekly infusion and it's intravenous. At the beginning, there are problems with young children to find their veins, and sometimes venous access is complicated and you need a portacath for infusion of intravenous treatment. This is also a problem for adults. Sometimes they get, it's too much for them and they do not comply their treatment. There is no adherence to treatment. As to hemophilia B treatment, factor IX concentrates are used, and each unit that is infused is going to increase factor IX plasma levels by 1%. Standard half-life concentrates last 18 to 24 hours, so prophylaxis is required twice a week. With extended half-life concentrates, the duration is 7 to 10 days, and therefore prophylaxis is required every 7 to 10 days. I apologize.
This led to the creation of extended half-life concentrates through different methods. One of them is fusion engineering to proteins, which is turning the coagulation factor into other molecules or substances. By way of example, in the case of this technology, which is the technology involving the fusion of the crystallizable portion of IgG, the crystallizable fraction receptor binds the protein. In the case of factor VIII, the recombinant factor VIII, they get bound, and as a result, it doesn't get degraded. And therefore, it has a longer half-life. They have appropriate safety levels. And in this case, for example, we have the binding between factor VIII and Fc. They are the two products that eloctate and eloctate, and the factor IX linked to Fc, it's alprolix. We see here the fraction bound to factor VIII, and we see that this results in greater stability and the longer half-life. In the case of albumin, it is a human protein with a long half-life in and for itself. It's a quite stable molecule with a prolonged half-life. So the recombinant factor IX was bound to albumin, and thus we prolonged the half-life, thus avoiding degradation. All of these drugs created through fusion engineering have both been approved by the FDA and the EMA. We also have another method to obtain extended half-life factors. This method is pegylation. It consists in a factor manufacturing process by binding the factor to the polyethylene glycol molecule. Binding the polyethylene glycol, PEG, to the factor VIII or factor IX protein, thus resulting in a reduced reduction in the interaction with clearance receptors, thus extending the half-life. There are several sites to which this PG molecule may be bound to obtain pegylation. One is in factor VIII at the cysteine mutation site. That's where the factor VIII molecule would be placed. That's the GV, and it's been approved by both FDA and EMA. We may also be working with pegylation, and in this case, this binds to coagulation factor. I mean, sugar molecules get bound to coagulation factors. Here, the sugars are attached to coagulation factors. In this case, it's factor VIII, and we have, in this case, Revinin and Refixia, both manufactured by Novo Nordisk, and thus reduces degradation and increases the half-life. Both have been approved by the FDA and the EMA. There may also be random pegylation. Here, as you see, is random. It can be attached to any site. I mean, the factor VIII molecule can attach to any site. The product in this case is adenovate, and this prevents degradation and extends the half-life. It has also been approved by FDA and EMA. There are other modifications in factor VIII recombinant factors. This is to achieve greater affinity for the von Willebrand factor. The von Willebrand factor serves different roles, one of them acting as carrier for factor VIII. So if we have a factor VIII, a single chain with a covalent linkage of heavy and light chains, this is attached to the factor, thus obtaining increased affinity and extend the half-life. This is Apsara. Also, we have a modification in the glycosylation and sulfation pattern, thus extending the half-life as well. This is the case of Cimoctocog alpha. The commercial name is Nuic. These drugs have been approved by both FDA and EMA. Going in greater depth into F-morococtocog alpha, Elocta, and Eloctate. This is a recombinant factor VIII with deletion of the B domain linked to the Fc fragment of IgG1. It was the first extended half-life factor VIII, and this half-life is around 19 hours. In many studies conducted, it was proven that people with hemophilia A have a low ABR between 0 and 2, with weekly doses averaging 1.8 to 2.4. These studies reported no development of clinically significant inhibitors. In studying these drugs which have been bioengineering treated, we measure PK, what is the clearance time, the time it takes to reach a trough, and also the clinical form. And how do we look at the clinical form? Well, by taking the ABR, annualized bleeding rate. This is a way to measure treatment efficacy. This is a summary of pegylation, which is a covalent binding to pegylated molecules, thus reducing glomerular filtration. Also, proteolytic degradation and clearance are reduced, translating into an extended half-life. Also, there is the Fc domain fusion of IgG or of albumin into protein. The neonatal Fc receptor is located in endothelial cells, thus facilitating protein recycling on the cellular surface. for subsequent release into the bloodstream. Also, there may also be glycosylation, and this translates into increased enzymatic stability, reducing renal excretion because of an increase in molecular mass. There are new molecules which are undergoing stage 3 trials that use X10 protein polymers. The mode of action is incorporating non-structured polypeptides in order to extend the half-life. This is used for factor 8. Also, we have the development of a carboxyl terminal peptide where there is a fusion of the peptide. The peptide is CTER into chorionic gonadotrophin, and there may also be a modification of amino acid sequence, which improves stability. And this is used in the design of one-chain recombinant factor 8. All these with the underlying idea of improving the half-life so that fewer infusions per week are required, which translate into fewer IV infusions a year. Among the latest molecules, we have efanesoctocog alpha. It is a new treatment option with the replacement of factor 8, which uncouples recombinant factor 8 from the endogenous von Willebrand factor. If you remember, I told you this was the carrier, thus increasing the half-life. It is formed of a single RF8 with, because factor 8 domains do not have a B domain, and it binds to the Fc fraction, to the von Willebrand factor here, and also to the X10 complex. All of these molecules cause factor 8 to avoid degradation and clearance, and thus extends the half-life. This is a trial, a phase 3 trial involving 159 patients. It's a multi-center study with patients with severe hemophilia A, patients above age 12, by changing prophylaxis and using this new molecule, efanesoctocog alpha. The annualized bleeding rate went down from almost 3 for those who use the standard half-life factor to 0.69 using this new molecule. So there is a 77% reduction in the ABR. Also, it was noticed that prophylaxis 1 time a week, 50 units per kilogram, gave an average factor activity greater than 40% for approximately 4 days after administration. And on day 7, there was still a 15% factor 8. No inhibitors were developed. And the half-life increased to 47 hours thanks to this modification. This is a summary of the different molecules. We have the one for eloctate, which is an Fc fusion protein bound to a recombinant factor 8, and the plasma half-life is 19 hours. So it increased by 1.7 times. The half-life increased by 1.7 times. As to Alprolix, which is a recombinant factor 9 bound to the Fc factor, the half-life was 82 hours, so there was a 4.3 time increase in half-life. As to rurioctocog alpha pegol adenovil, which is a pegylated product, the half-life is 14.3, and it was prolonged between 1.3 and 1.5. Then Ildevion notice the plasma half-life 100 times. Then nano-cog beta-pegol, Refixia is the trade name. The modification technology is glycopegylation. It has a 93 plasma half-life, and the half-life was prolonged by almost 6 times. The moctocog alpha-pegol, GV, that's the commercial name, is a pegylated product. It's a factor VIII pegylated product. The plasma half-life is 19 hours, and the half-life was prolonged by 1.6 times. Turoctocog alpha-pegol, the trade name is Esperoct from Novo Nordisk, is a glycopegylated recombinant factor VIII, achieved a plasma half-life of 18.4, so that the standard half-life was increased by 1.6 times. This is saying more or less the same, the prolongation of the factor IX half-life. And here you have the treatment of hemophilia A with extended half-life factor VIII concentrates. For pegylated recombinant factor VIII and Dinovate, the recommended dose is 45 units per kilogram, twice per week, and the average, the half-life is 14.7 hours. The pegylated recombinant factor VIII, GV, the dose is 30 to 40 units per kilogram, also administered twice a week, and the half-life is 18.7 hours. Yet another recombinant factor VIII, pegylated recombinant factor VIII, Esperoct, the dose is 50 units per kilogram every 4 days, and it has a half-life of 18.4 hours. Eloctate is used at a dose of 25 to 65 units per kilogram every 3 to 5 days, with a half-life of 19.7 hours. Astila, which is a recombinant, a modified recombinant factor, the dose is 20 to 50 units per kilogram 2 or 3 times a week, and the half-life is 14.2. And Nuwic, which is a recombinant factor VIII, also glycosylated, modified factor VIII. The dose is 20 to 40 units per kilogram every 2 to 3 days, and the half-life is 17.1.
As regards hemophilia B, treatment with extended half-life factor IX concentrate, what we have available is Alprolix, which is a factor VIII recombinant with Fc fraction, that is 100 units per kilogram, 7 to 10 days, and half-life 82 hours. Then Ildevion is used with 71 units per kilogram every 10 days, and the half-life is 101 hours. And recombinant factor, pegylated factor IX, the dose is 40 units per kilogram every 7 days with a half-life of 93 hours. As we can see, this has allowed going from infusions 3 to 4 times a week, that is 12 to 16 times a month, multiply that for the year, and this is intravenous. This, in the case of hemophilia A, will be reduced to twice a week. So there is a significant reduction in the number of infusions per week. And this contributes to improved treatment compliance. And in the case of hemophilia B, even better, because from 8 times a month, because with standard half-life factors, prophylaxis for hemophilia B is twice a week. But with these factors, with extended half-life factors, this is usually every 7 or 10 days. So instead of twice a week, it's going to be once a week or once every 10 days, 3 times a month. And this results in an improved quality of life and improved treatment compliance. I have brought a study we conducted in our center with extended half-life factors. We are part of the humanitarian aid program with the World Federation of Hemophilia. And in 2019, this pharmacokinetic study was performed, and we included several patients with hemophilia A and B, 153 patients with hemophilia A, 37 patients with hemophilia B, aged 2 to 68 and 13 to 74. This is the dose. The PK study was performed in 34 patients who used Eloctate, that is factor VIII. And we saw that average half-life was 21 hours with a range of 11 to 43 at an average dose of 40 units per kilogram. And in order, the time for trough level was 125 hours, where you obtain the lowest level with the dose administered. So the trough level was achieved at that point, so there was a significant protection time. In PK studies, they were performed through the WAPS-HEMO system developed by the McMaster University in Canada. And we have access to that through this website. So centers can communicate with this university and have access. So PK studies have to be conducted with taking blood samples before administering the factor, half an hour later, 1 hour later, while in the past, PK studies would require 10 to 12 determinations, while with this system developed by WAPS-HEMO, with 3 determinations, we managed to have the projection, the PK projection. So treatment can be customized because not every patient responds in the same way. In children, you need a faster factor clearance. You need to see the standard half-life in order to adapt it. For instance, if in a patient it was an average half-life of 21 hours, we can use it every 5 days. While if the average half-life is 17 hours, so we will use it every 4. So we can do something more customized. In the case of factor IX, 16 patients, average half-life was 81.7 hours with a range of 62 to 130. The average dose used was 21 units per kilogram. The range was 10 to 49. And the time to reach the trough level was 208 hours. So we see that by using the average of only 3 determinations, it was possible to estimate the PK on an individualized basis using the WAPS-HEMO system. And we could see the difference between extended half-life factors and standard half-life factors. In the case of factor VIII, it was 1.3 to 1.8 times greater than other products. And in the case of factor IX, it was 2.5 times higher. And time to reach trough was longer in extended half-life CFCs. So as explained before, this allowed adjusting the prophylactic schedule to each patient. As regards factor 8 FC, the frequency is usually once a week. Is usually children. This changes if the annualized the ABR increases to 1, then we will increase the frequency to twice a week. PK helps with this, but clinical evaluation is extremely important. In patients where prophylaxis was applied every 5 days, that is 61 patients, twice a week in 62 patients, and 3 times a week for 11 patients. As to factor 9, it was every 7 days in 27 patients and every 10 days in 10 patients. By way of conclusion, we may say that in these patients, the use of recombinant factor 8 bound to FC fraction and recombinant factor 9 bound to FC fraction proved to be a safe and effective therapy. We can also say that the PK estimation model that we used is quite a simple and available tool that helps clinicians optimize prophylaxis for hemophilia patients. The use of these extended half-life concentrates has been very beneficial to our patients, especially those with hemophilia B, where we managed to reduce the weekly frequency of infusions even when using low doses. This is the staff at our National Hemophilia Center. This is the multidisciplinary team. I thank you for your kind attention. And we are celebrating the World Hemophilia Day, that is tomorrow. This was 4 years ago when we took a journey to celebrate World Hemophilia Day to a mountain close to our city of Caracas. These are patients and part of our multidisciplinary staff. Thank you for your kind attention. And if you have any questions, I am here to answer your questions. Thank you. This was indeed a very interesting presentation. Now we invite participants to write their questions in the chat. We also have the questions that were presented when people registered. First question from Denise Lopez from Colombia. She's asking, what are the benefits or consequences from a standard half-life to an extended half-life factor? And is there any contraindication in the change of molecule? Well, very important question. Switching has been done and studies conducted specifically measuring the switch from standard half-life factors to extended half-life factors. We did not see one of the most feared complications, which is the development of inhibitors. There wasn't an increased inhibitor development, not higher than usual, which may usually be related to other problems. So we saw these are safe, and there was no problem at the time regarding the development of inhibitors when moving from standard half-life to extended half-life. The big advantage to this is that they can be used at a lower frequency. Thank you. Second question from Edwin Vendesu from Peru. What are the probabilities to acquire inhibitors in pediatrics? Same thing. Remember that inhibitor development is a multifactor situation where there are endogenous factors, including the type of mutation. There are some molecule mutations, both of factors 8 and 9, when there are peak deletions of the molecule. This will foster the development of inhibitors. These are factors. Also, ethnic origin You know that African people have an increased trend to develop inhibitors. And this has happened with several factors that are endogenous, but some can be modified. For example, the use of high dose treatment with very young babies because of a surgery or a hematoma of the brain. A continued dose may contribute to inhibitor development. The development of inhibitors is quite complex, but when we switched, we did not see increase in inhibitor development. There is no problem in that regard. Did I make myself clear? We have one more question from Hugo Reyes asking whether the Colombian health system has been informed about the benefits of this molecule. We can find out. As far as I understand it, correct me if I'm wrong, this is a type of treatment that has been available for several years, and most likely the Colombian health system is aware of it. What we do not know is whether it is available exactly. Each country has their own rules, and I wouldn't know. Most likely it is available, but I do not know whether it's being distributed. I couldn't tell you because I'm not familiar with that. We have another question in the chat from Lucas Ruben Figola. He's saying, excellent presentation. I have a question. In clinical trials, have you done a clinical trial taking into account men and women with hemophilia individually? Have trials included both men and women or just men? And if they were specific for one or either gender, has any study been developed to be precise in women patients with hemophilia and how they are affected, as well as the level of inhibitor development? Studies have been conducted with men, and let me tell you why. In general, in the severe phenotype affects women less than men because they have the other chromosome that somehow protect them. So there are less women with a severe phenotype. Severe phenotypes are more frequent in men. Therefore, prophylaxis studies have generally been developed in men, in male patients. This is different from the case of von Willebrand, which is a separate bleeding disorder. And there, the distribution between men and women is similar. This affects both sexes, both genders. So in that case, the use of prophylaxis in persons with severe von Willebrand is quite convenient because in women you can avoid problems with their periods that are so heavy. They have heavy periods and they spend 10 to 15 days with their period bleeding. So this worsens their quality of life. So prophylaxis in hemophilia, although there are women with hemophilia, even if it's less than 40, it's hemophilia, but in most cases, it is a mild phenotype. And in hemophilia A, desmopressin is used for mild hemophilia in women. Also, factor 8 can be used, or factor 9 can be used in women, but for some circumstances, for instance, for surgeries, or when they go to the dentist, or if they are pregnant for their delivery or C-section, for those special situations.
This is a question from Luis Rojas. It was me. You read it, go ahead. The first one is from Luz Rojas, which says, the slide says that the half-life is 82 hours. That half-life is for any amount of units or just 100 units per kilogram, talking about Alprolix. In the study I have presented that was conducted at the National Hemophilia Center, the hemophilia had a range of 100 to 60. She corrects herself, 10 to 60. We did not reach 100 units per kilogram, and however, the half-life when measured, the average was 21.7 hours. That is why, ideally, we should have the PK studies, because then you can say, well, okay, this is the half-life, so we will do it every so often. Usually it's difficult to do this in all patients, but at least you have a picture of what the performance or response was amongst our patients to these drugs. And based on that, the decision was made to do it every 7 days. But nonetheless, the clinical portion is very important. That's why patients should visit their physician every so often at a regular interval. You keep records, you check the joint, you see what the bleeding rate is. That ABR is very important to check how prophylaxis is working. Another question from Ecuador. How important is the role of the lab results to determine the development of inhibitors in molecule exchange on an ongoing basis, especially from plasma to recombinant? That is, determination is very important and basic for follow-up. And what is the solution to know what the Bethesda units are? Well, thanks for the question. Lab is very important. In the case of inhibitors, we have to think, patients themselves or children's mothers are the ones who tell us, but it is children's mothers who tell us, well, I see that the child is not responding. We administered the treatment, but he still has, or she still has bleeding. So this is a red flag. On top of that, in the case of young children, whenever the treatment starts, especially prophylaxis, starts 5 days after the child started receiving treatment, that is with 5 doses, we need to determine what the inhibitors are, and the same thing 20 days afterwards, and then after a month, then every 3 months, every 6 months. And in the case of adults, this is done once a year, or if they need to undergo surgery. Why is that? Well, because they may have developed inhibitors. But if a person has not developed inhibitors 150 days after being exposed to the treatment, this is hardly ever developed afterwards. In the case of children, it is important to be paying attention to inhibitor development. Not only because there may be a switch from one factor type to the other. This has to be done on a routine basis, even if they receive standard half-life concentrates or extended half-life concentrates. Determining inhibitors is always important. I have 2 questions. In Uruguay, all hemophilia B patients are receiving treatment with the standard coagulation factor, and the 2 drugs that you mentioned are now being registered. So the switch is immediate, or does the patient need to have some kind of low dose, or what happens or what you do when you administer other treatment options? And the other question is whether these factor types coexist with the standard one when necessary or not, or whether they have served the purpose. In the case of emicizumab, the emicizumab coexists with a factor. What is the case here? So it's different from emicizumab, because remember that emicizumab is a non-replacement treatment. Emicizumab is used only for prophylaxis purposes, not in the case of bleeding. That is why if the person has had a tooth removed, we need to apply factor VIII. In the case of inhibitors, you will have to bypass it. That's as an aside. As to the switch from the standard half-life factor to extended half-life factor, no preparation is required. Simply, you need to be more alert because any change that the person may represent to have is important, but generally there are no issues. However, we need to pay attention and do a follow-up on the ABR, on the development of inhibitors, and it's also convenient to test for inhibitors every 3 or 6 months and be paying attention on the product type. We normally recommend what the best test or lab method is to determine both the factor and the inhibitors. You have to be paying attention to that, but it is the lab that is responsible for that in general. However, no special preparation is required. You can say, well, until this date, he has used this, so starting with the next dose, it will be this other treatment, and we will go with the tide. And as to the coexistence with the factor VIII, well, no, the coexistence, that other question. Well, what happens is that if it is already using the extended half-life factor, well, try to be successful with that one. Let me say this specifically, and apologies for using the Venezuelan case, but I work here. As I have explained, extended half-life factors were donated by the Federation through the humanitarian aid program. The authorities here, the Social Security Company, are acquiring Nuwick, which has an extended half-life shorter than Eloctec, but it is extended half-life nonetheless. So what happens? We try to use what the Social Security provides. The request was put so that all patients would have this, but sometimes distribution fails, and if the other products are available, we do use them. It's not ideal, but so far we haven't had any problems. Ideally, if the person is receiving a given factor, he should be receiving that. Here, we have the issue of drug procurement perhaps not being sufficient to cater for all patients. Well, thanks. Thanks very much for the presentation. It's been very good. Thanks to all those who participated. I don't know if there's any other question. Otherwise, we will be closing this last meeting, and we are going to celebrate with the World Hemophilia Day tomorrow, raising awareness, sharing knowledge. So thanks for your time. Thank you for the invitation to participate, and let's celebrate tomorrow. All of us who work with you, and you, of course, have to celebrate as well. Thanks very much. Have a good evening. See you soon. Bye-bye. Thank you.